Well, welcome back, and I'm Steve Boppart, and it's my pleasure really to introduce the presentation that Adeel Ahmad is going to give in a second. Adeel has been with my group for 5, 6 years? 4 years. It seems like a long time. 7, 8. But he's been doing this incredible work. He comes with from this ComSats program, a fellowship that got started here. But he comes from electrical engineering, where with a lot of expertise in signal processing and imaging, and is applying that now to more broader topics of nanotechnology and using nanotechnology in interesting ways to sense biological systems. And so one of the things that both he and I have had to learn and relearn is that when we start using these nanoparticles, and in particular this type of nanoparticle that is motile, is that it's moving, we've had to learn a lot of mechanics. And I think that's what's been exciting for me and for Adeel as well, is that we've had to learn a lot about tissue nanomechanics. So Adeel is going to go into a lot more detail. This is just one slide to introduce this concept, and it's a concept we developed a couple years ago in my group called magnetomotive molecular probes. And these are magnetic nanoparticles that can be dispersed through tissue, targeted to tissue. When we apply a very small, weak modulating magnetic field, they actually move. And they move on the nano to micron scale. And we can do all sorts of things with these. We can use them to enhance contrast. We can use them to sample the mechanical properties of the tissue. Because we can pull on them, let them go, and see how they respond, and that's dependent on the mechanical properties. And we can actually even target these, and in some instances, turn up our magnetic field strength and frequency, and they heat up, so they can be a therapeutic application. So I'll pass this off to Adeel, and he'll go into more detail about an interesting use of these, combining them with microspheres and some of these other applications.
Yep. Thank you, Professor Boppart, for the introduction. So today I will be talking about our group's ongoing works in using multimodal magnetic So in today's talk, I'll be talking about our group's ongoing work in using multimodal magnetomotive microspheres for cancer imaging. So here's a brief outline of my talk. So first I'll talk about the structure and the preparation protocol that we use for synthesizing these microspheres. Then I'll talk about some of the experimental methods that we use to externally modulate these microspheres and to measure the small nanoscale displacements within tissues and cells. Then I'll share some results with tissue phantoms and with a rabbit atherosclerotic model and a rat tumor model. Then I'll discuss some ongoing research and future directions that we are taking with this work. So the basic motivation behind this work is to develop particles that can be targeted to different cellular receptors and then subsequently be used for in vivo imaging and for therapeutics purposes. So in this case, our microspheres contain iron oxide nanoparticles inside, which are magnetic. So the advantage of using magnetic particles is that they can provide imaging contrast across multiple imaging modalities. For example, MRI and magnetomotive optical coherence tomography. These magnetic particles can also be externally modulated, and the displacements can give an idea of the surrounding mechanical microenvironment. And another advantage of magnetic particles is that by applying a high-frequency magnetic field, these can be heated up. And this can be used for therapeutics and controlled drug release delivery applications. So here's the basic structure of the microspheres. So these microspheres that we use have a core and a shell structure. So the core is hydrophobic, consisting of vegetable oil, and the shell is hydrophilic, consisting of a protein bovine serum albumin. The flexibility of this structure allows us to incorporate different materials within the vegetable core. So in this case, we are using iron oxide magnetic nanoparticles, which are roughly 20 to 30 nanometer in size, and a fluorescent dye, Nile red. What the iron oxide allows us to do is to generate contrast across multiple imaging modalities, for example, MRI and magnetomotive optical coherence tomography. And with the Nile red inside, we can do fluorescence imaging. These microspheres also generate contrast in ultrasound imaging. So these microspheres are prepared by high-frequency ultrasound. So here is the apparatus. It basically consists of a titanium horn and a glass vial immersed in a temperature-controlled water bath. So the solution to be incorporated in the microspheres is placed in the glass vial, and the tip of the titanium horn is placed at the oil and protein interface. On the application of a high-intensity ultrasound, emulsification takes place and protein aggregation occurs at the oil-water interface, which results in the cross-linking of the proteins, which form the shell around the vegetable oil. Further steps include purification and washings. So once we make these microspheres, we need to target them to different cellular receptors. So as a proof of concept, in this case, we are targeting these microspheres to the alpha V beta 3 integrin receptors that are known to be overexpressed in tumor vasculature and arteriosclerotic lesions. So the way we attach the RGD tripeptide is by a layer-by-layer electrostatic adhesion process. So the outer shell of the microsphere is negatively charged, consisting of a bovine serum albumin. So we attach a positively charged PDDA layer, followed by a negatively charged silica layer, and then attach the RGD peptide sequence to this. So to measure the microspheres or the magnetic response of these microspheres inside the tissue, we use an optical technique called optical coherence tomography. Optical coherence tomography is a real-time, non-invasive imaging technique, which in principle is very similar to ultrasound, but it can give images at much higher resolution than ultrasound, but at a lower penetration depth. So in optical coherence tomography, the laser light is split into two arms, the reference arm and the sample arm. The light in the sample arm goes inside the sample and is backscattered from different structures within the sample. So the backscattered light combines with the light in the reference arm to form an interference pattern, which is practically dispersed by the grating and is focused onto a line scan camera. And after some further processing, an optical coherence tomography image is formed. In order to detect the magnetic particles with OCT, we modify the OCT sample arm by placing a solenoid coil. So when we apply a magnetic field, the particles will respond to this magnetic field and will start moving. So this is a cartoon of the OCT sample arm in which we place like electromagnetic coil. And if the sample contains magnetic nanoparticles or magnetic particles, on the application of the magnetic field, they will respond to it. The force generated by the magnetic field will be proportional to the volume of the particles, the magnetic susceptibilities, and the magnetic field gradient. And as these particles will move, they will change the amplitude and the phase of the interference gram. And OCT can detect this change in the amplitude and phase, and based on that, we can detect the presence of the magnetic microspheres. So this slide basically just outlines the processing steps that are used to form a magnetomotive image. Basically, it includes taking a transverse Fourier transform and bandpass filtering the modulation frequency. For example, this is a Fourier transform of a sample that doesn't contain magnetic particles, and you see nothing at the modulation frequency. And this is a Fourier transform of a sample that contains magnetic particles, and you see a strong magnetic response at the modulation frequency. So you just bandpass filter this magnetic response and get your magnetomotive image. So this is a typical example of an MMOCT image, where the red is the structural OCT and the green is the MMOCT image superimposed on the structural image. So first we did a series of experiments using tissue mimicking phantoms to understand better the MMOCT signal. So in this case, we're preparing phantoms with different concentration of magnetomotive microspheres. So we see, like, with the increase in the number of microspheres, we see a corresponding linear increase in the MMOCT signal. So the MMOCT signal, in addition to depending on the concentration, will also depend on the stiffness of the tissue. So in this series of experiments, we vary the stiffness of the phantom by changing the percentage of the agarose inside the phantom. So we get a response like this. So in this case, if we use a very low viscosity sample, we don't get any MMOCT signal, the reason being that the sample doesn't have any elastic restoring force. And if we use a very high stiff sample, the magnetic particles don't displace, and again we get a low MMOCT signal. So this data suggests that these magnetic particles can also be used to assess the surrounding microenvironment. So in order to study the dynamics of these particles, we needed a very high-speed system. So we spent some time developing a high-speed system that does the processing using a graphics processing unit. So these are some representative data sets that we took. So this is a tissue phantom, and this was acquired at 75 frames per second. And this was a lung tissue, a rat lung tissue, again acquired at 75 frames per second. So in order to mimic like the blood flow environment, we used prepared phantoms with a capillary tube. So this is a capillary tube containing magnetic particles inside. And you can see it so you can see the flowing particles. And in this set of phantom, we have like a rotating magnetic field. And as the magnetic field rotates, you can see the magnetic particles rotating in response to the magnetic field. And what OCT allows us to do, we can do is quantify different information from this data set. For example, we can quantify the parabolic velocity profile using the OCT data sets. After characterizing the MMOCT signal from these magnetic microspheres, we did a series of experiments. So in the first set of experiments, we used a rat tumor model. And we injected these magnetomotive microspheres through the tail vein. After the injection, we did an in vivo imaging using ultrasound. And after sacrificing the animal, we did ex vivo MMOCT and fluorescence imaging. So these results are from the in vivo ultrasound imaging. So the green channel over here is the magnetic microsphere superimposed over the structural ultrasound image. The plot over here shows the scattering signal from the tumor location. So the x-axis is a function of time. X-axis is time. So at the tumor location, so we see an increase in the scattering signal, which indicates that the microspheres are accumulating in the tumor. So in this case, we use targeted microspheres that bind to the tumor receptors. In this set of data set, we applied a sequence of rupture pulses. And these after a sequence of rupture pulses, we see a corresponding decrease in the scattering signal, and which we believe is due to the rupturing of the microspheres. This indicates that these microspheres, if they incorporate different drugs inside if we can incorporate different drugs inside the microspheres, after the application of some ultrasound rupture pulses, we can rupture these microspheres, which can be used for controlled drug delivery applications. From the same tumor location, we excised the tumor and did MMOCT, where we can see the presence of these microspheres. We also did fluorescence microscopy on the tumor location. So the arrow points to the intact microspheres, and we also see like a diffused red thing, which we believe is the leaked dye due to the rupturing of the microspheres. So in another set of experiment, we use a rabbit atherosclerotic model. So the plaques also contain integrin receptors, and we believe like these microspheres can also attach to these integrin receptors because we are functionalizing them with RGD. So in these set of experiments, we took rabbits and we fed them a high cholesterol diet for a period of 6 to 8 weeks, so that they developed plaques. After sacrificing the rabbit, we extracted out the aorta, and for each segment, we divided that segment into three different segments. So we perfused one of the segment with targeted microspheres, the other with non-targeted microspheres, and the other with PBS, and that was used as the control. While we were perfusing the microspheres on the aorta, we did in vivo catheter-based imaging, and then MMOCT imaging and fluorescence confocal imaging. So this is one example of the extracted rabbit aorta. So you can see some branch vessels over there, and you can see some fatty streaks that contains the plaques. We built a customized flow chamber to perfuse the microspheres on the rabbit aorta. And we set the parameters so that they mimic the physiological conditions. For example, the blood pressure rate and the flow rate. So these are some representative images. So in the first column is the targeted aorta, in which we use targeted microspheres. So in this case, we see a very strong contrast in the MMOCT image, and this correlates very well with the confocal imaging and the histology. In the non-targeted aorta, in which we perfuse the microspheres that were non-targeted, we don't see any strong MMOCT signal. We see a very weak MMOCT signal, which suggests some non-specific binding. And in the control aorta, we don't see anything. These are some statistics based on these results, which indicate that there's a significant difference in MMOC signal between the targeted and the non-targeted cases. While we were perfusing the microspheres with the ureter, we also did catheter-based OCT imaging. So this is a representative video. So you can see the outer wall of the catheter, outer wall of the aorta, and you can see the microspheres flowing inside. So in summary, we've been successfully able to synthesize and target the microspheres to the integrin receptors in a rat tumor and a rabbit arteriosclerotic model. Some of the future work includes to develop an in vivo magnetomotive OCT catheter so that we can do MMOCT imaging in vivo. We also need to do more studies to test the targeting and biodistribution of these magnetic microspheres. We're also looking into encapsulating different lipophilic drugs inside the core of these microspheres for targeted drug delivery. And we're also looking at the possibility of using high-frequency magnetic fields to rupture these microspheres, again, for drug delivery applications. At the end, I would like to acknowledge all the people highlighted in the group that contributed to this work. Yeah, that's it. Thank you.
Time for a few questions. So what range of stiffnesses do you think you can explore with the, you showed how the...
Yeah, so right now we just like to change the concentration of the agarose gel.
Right.
So like I don't have the quantified number, like how do they quantify to the actual stiffness at the moment? Yeah.
But in that experiment you were showing that it does respond to the stiffness of the...
Yeah, so at low, so the magnetomotive principle like requires a restoring force. So at very low viscosities, you don't have a restoring force that can pull like the magnetic particles back. So you don't get any MMOC signal. At very high stiffnesses, you have no movement of the magnetic particles. So again, you get no MMOCT signal.
What portion of your magnetic particles or the particles that you introduce do you think actually get targeted to the place where you want them to go? You do tail vein injections. A lot of that ends up in the liver a lot of times.
Yeah, it does. Like especially like in the normal rat, most of them will end up in the liver, the kidneys, and the bladder.
And what portion do you think go where you want them to go? How much do you think you could target directly?
Like I don't have really the numbers, like how much, like you're asking like how much percentage will go into the tumor. Like I would say like roughly, like I don't have the exact numbers, but I would say like even in that case, you still see some accumulating in the liver and the kidneys, but like roughly like 10 to 20% would go into the tumor.
So what do those particles do to these other organs?
No, so in this case, what we're using is microspheres. So the microspheres are roughly like 2 to 5 micron in size. So they remain like in the tumor vasculature, for example. They remain in the vasculature. So they won't go like inside the tissues.
Oh.
If we use like nanoparticles, so they would probably diffuse inside. So these are really like blood pool particles that would remain in the bloodstream, just because of the size.
Any other questions? Okay, all right, let's thank you for speaking.